Brilliant. Sorry for all the
Okay.
interrupting. The stage is yours.
Okay, great. So let's keep it informal just because we're not many, and I think at least let's keep it informal and interrupt me whenever you like, and let's have a discussion about shape in biology. Recently, I gave a public talk to the Department of Physics, and I prepared a slide which I think is useful today. I'm gonna make myself smaller. Okay, it's useful today because you're students of physics and you say, why is this woman doing biology in the Department of Physics? And what does it relevance have with me? Well, I'll tell you, I think all the relevance. In fact, I think we've been obsessed with biology from the beginning of physics in the 17th century. It's just, we couldn't do it. And I'm going to show you how underlying a lot of physics in the 20th century, which is where our favorite physics stands, is this thing of understanding where are we, why can we understand the universe, why experiments coupled with our intuition and logic in mathematics can lead us to some discoveries of truth, whatever that truth is, and how that truth is usually translated into technology. If you realize that's what as physicists we do. We try to discover some kind of truth in the universe that it can help us to understand how the universe works. And usually with that knowledge, one of the ways we prove the universe works in the way we think, that our mathematical models are correct and that our intuitions to reach those mathematical models were correct, is by building technology. Now, you know, physics starts 17th century with the invention of the microscopes, the telescopes, Galileo. The first thing people look at, Robert Hooke in Oxford, Leeuwenhoek in the Netherlands. What do they look at? They look at life. The first thing Robert Hooke looked at was the cork. He discovered cells, and Leeuwenhoek discovered microbes, and actually was the first one, and this is current biological physics, to discover the movement of a sperm. Interesting things he was looking at the first time he used a microscope. And he actually made a first biophysical model of how a sperm would move. The propeller, we just learned is wrong a couple of years ago. Some biophysicists learned it was work, but it's not just the beginning of instruments. So the first instruments that would allow us to understand the universe, the microscopes, the telescopes. The is most of the development of physics on the 20th century somehow leads us to biology, because the biggest mystery of the universe is why the universe creates life and why life like us is able to understand the universe. And this has been central to physics from start. Maybe I'm taking a bit long with this, but maybe it's useful as an undergrad to think about that. Also Galileo, the first time he was looking at the sky with the telescope, one of the first things he had to prove is that the information coming through this telescope was actually not a distortion of reality, that it was actually the pictures from the telescopes were the pictures of the sky. So always this interplay between our intuition, our instruments, our experiments, and logic leads us to physics. Okay, so let's go through 20th century physics. Beginning of the century, we're all worried about the quantum nature of matter, atoms, quantum mechanics, and then of course general relativity in the other extreme. Those things that still haven't come together, and I come that at the end of my talk. So as soon as we discover atoms, people discover X-ray crystallography, we start to get with things that are more complicated. As soon as we get to condensed matter physics, people start to develop models such as Lev-Landau, etc., of things emerge, superconductivity. So from atoms, immediately we learn that as matter becomes more complex, even simply complex, like a crystal, we start to, the effort is always to understand what does it mean to become more complex. And what it means to become more complex is that the sum of the parts is not the same as the total, and that funny things happen, like superconductivity that you don't immediately predict from the atoms, and that to reach those theories, you need to coarse-grain reality. You don't take into account single atoms in order to create solid-state physics. Solid-state physics is a very clever thing that puts together a lot of fields of mathematics and physics to create theories that predict macroscopic behavior in a way that is not clearly, so it's like emergent behavior. Complex systems start to create behavior that does not resemble the behavior of single atoms. As soon as these guys, or at the same time, these guys were working in condensed matter physics, the development of technology, the invention of the transistor, of course, the transistor links with mathematics, with the origin of computation. And computation is something biological systems do. Computation is about having an algorithm. Can I know the solution of a system? Can I create something intelligent? At the same time, people like Lars Sonsager were trying to understand things out of equilibrium. Most of the 19th century thermodynamics developed by was concerned or could only solve things in equilibrium. But people start to think in complex systems and start to think like, okay, so why do we have things like life in the first place? Or how come, this was Schrödinger in 1944, starts to think about this problem. So if entropy grows, why the universe creates something as complex as life? So basically the answer to this from a thermodynamic point of view, from Lars Sonsager, Ilya Prigozhin, is that you can decrease your entropy if you dissipate energy into the environment. That brings the beginning of, was the origin of life actually a system that was able to dissipate energy and reduce entropy? In the moment you reduce entropy, and this is something I want you to understand, when you have some system that through interaction with the environment is able to reduce its entropy, what it means is that it's creating a shape. That shape has information about the environment, where it comes from, the way it dissipates energy into the environment. All that creates a shape. That shape is related to that entropy. And this is something that Claude Shannon understood in the 1940s as well, at the same time as the transistors were invented. Now, here I have more physics, at the bottom I have more mathematics. Claude Shannon was interested in information. Why? Because since the beginning of the 20th century, mathematics, at the same time as physics was interested in shapes, in atoms, in matter, mathematics were interested in knowledge. So, and how we acquire knowledge, was interested in logic, the limits of our logic, and what could be computed using logic. And let me bring two of the heroes of this story. Gödel's incompleteness theorems, 1931, following the work of Cantor on foundational mathematics. So Cantor is the one that looks at why is infinite and why can we think about it. And then Gödel realizes that in a system, in a logical system, in which we create a set of rules that we believe they're true, and then we follow the rules, you're going to end up with computations of that system. You're going to end up with things that come in that algebraic system that are true, yet you cannot prove they come from the actions you define in the first place. So there's things outside logic. This is the most interesting thing I'm going to bring you here when you do computations. Now, of course, Turing shows that a lot of things are computable, and he creates the Turing machines. And then for Neumann, it starts the world of computation in a more serious way. All the computers we use for Neumann architectures. And Claude Shannon is the one that realizes this link between thermodynamics and information is entropy. I'm going to go back later because it's the idea that in a shape, in the reduction of entropy, there is information. The idea that storing information from your environment by dissipating energy into the environment leads you to a storing of information. And later I will tell you how that leads to computation. So of course, this follows in many ways, and at the same time, physics starts to use computers. So now we have 1947, the development of transistors. These guys in the 1940s start to realize that we can compute, we get the computers, and by the 1960s, people were already using computers such as Lawrence to understand complex systems. What happens when you have a lot of things coupled together, such as the weather, such as a flock of birds, a couple differential equations, and you let them evolve over time. So now you could start to solve those problems. That's the beginning of chaos theory. And indeed, in the 1960s, something really clever, soft matter physics. So Pierre-Gilles Lejeune starts by just arguments of fluctuation of polymers, soft matter. We come back later about what is the meaning of soft matter and why life emerges from soft matter in the first place. But he realizes that using arguments from statistical mechanics, one could intellectually, sort of using physics, breach the nanoscale of the polymer with a macroscopic behavior. Of course, I jump here over the only woman in the picture. Photograph 51, Rosalind Franklin discovers the structure of DNA. And again, another application where I started at the beginning. Physics have always been involved and trying obsessed with life. We're interested in complexity from the beginning. We're interested in computation, the meaning, the relationship between emergent phenomena, complexity, and computation have been there throughout all the motivations of physics in the 20th century. And of course, this is a groundbreaking paper that is very famous in the people interested in complexity theory. Complexity theory starts here, the idea that when you have computers, you start to be able to model more complex systems. And that complex systems, going back to superconductivity, when you have things strongly coupled with each other, give rise to emergent phenomena. And this is what he summarizes in this paper that it was groundbreaking. I think recommend you to have a read of it. It's very good. It's called More is Different, Broken Symmetry and the Nature of Hierarchical Structure in Science. The idea that when you build up structures in a hierarchical way, which we all are, you end up having a lot of interconnections between them, a lot of feedback loops that end up having emergent behavior that is at the origin of the complex behavior we see at the macroscopic scale. So this is the beginning, if you want, of biological physics. And in the so I started working in this area in the 1980s. So using atomic force microscopy, I started to look at molecules at the nanometer scale. At the time, I was just really an atomic force microscopy. I didn't have much thinking of physics. There was not. I was just trying to help biologists to understand their systems, but slowly it all dawned on me that it was all physics there, and that I could use the tools of nanotechnology to start to use some of these ideas I was telling you in the previous slide. Now, let's go back to this idea of shape and physics and environment and why a biological molecule can compute and learn from the environment. During the pandemic, I've been walking around in the morning near where I live in the Aston Yacht. And there's been a lot of frost, and I got quite obsessed looking at the structures of ice in the water and the life, and thinking a lot about this idea of what type of structures on the universe are able to endure time, and what allows you to survive. What's the physics allowing us to survive over time? So the things that last a long time are hard things like crystals, rocks. They're really last for a long time. They're really they're really elastic things. Elastic means that response instantaneously to mechanical perturbation. They don't dissipate energy into the environment. They just stay there. They're eternal. For them, there's no time. The time comes in hard structures from the interface with the environment. So in ice or in steel or whatever you are, you will stay there forever. In the case of ice, if the temperature is low enough. And the only interaction you have with your surrounding is always atomic. And this is very interesting. So even if you're ice, it's just your atoms that are getting information in and out. And the same with the steel. I will go back later to steel. The only steel usually doesn't last in time, not because it's not hard enough, but because it corrodes. So you will have chemical reactions happen as an interface, oxides developing, and actually the steel can break. And what you usually do, and this leads to actually the talk that Paolo Radelli will give tomorrow in the department about the capacity of oxides to compute, which is very interesting because I think it has a very interesting convergence with the origin of life, which is the idea that when you have an oxide, you already have something soft. You already have something that is able to rearrange in the environment over time, coupling chemistry with mechanics, with the rearrangements of the oxide, at least in a steel. And I think this is the reason why engineers and architects use something called Corten steel, which is a steel that they on purpose oxidize at the surface, and that oxide allows the steel to last forever. Because that steel, by rearranging itself with the environment, is producing kind of primitive computation that allows the steel to stay over time. So if looking at a steel, I find, which is a good way to understand why life emerges from soft matter, is the idea that when you have a softer structure, like a polymer, here I'm having a string of DNA, look at it with AFM, I will show you, and they're always fluctuating. These are stuck to the surface, but they're coupled to the environment. A softer structure coupled in environment is able to couple its shape to the environment. So basically it's coupling the information he gets from an environment to his entropy, to his shape. And this is very interesting because we go back to this idea of Claude Shannon of linking thermodynamical entropy with information. The idea that when you're soft and you endure time, going back to the idea that you persist over time, you are storing information in your structure. But that's not enough. If you only have a polymer, it won't be enough to survive over time because eventually there will be something that will uncoil. That's the origin of life. How the first molecules started to create structures that would allow them to endure over time. We're still here 4 billion years after life keeps going. And you will see, and we think, is because there is a computation involved. You need to extract information from the environment and do something with that information that allows you to keep alive, collect food, change your shape, and allows you to move and eventually to replicate. And that's the physics we're trying to understand right now. So this is very interesting physics. We'll come back later to that because it's really at the interface of so many fields right now in physics and the sciences is really the cutting edge. The idea of what is computation, what can we compute? I come back to the end in the lecture to the quantum computers. What is computation? Why we need shapes to produce those computations? What is the in the case of computation, the shapes of the algorithms, how those do relate to the shapes of biological systems. As you know, probably neural networks mimic the structure of our networks in the brain. And it brings us to fundamental problems about the origin of life and even about the, as I come back later, of when does quantum computation become something classical in the real world. So I'm going to take you to some examples of my work and something that is not my work. Actually, there was a paper that came out yesterday in PNAS, and I decided to bring it to you because it's fascinating and it's a good complement to my own work. So I work on I try to understand what makes, how can I experimentally measure the physics of something that has a shape, like for example, a plant. So the different shapes, so plants don't move very much. Well, they move their leaves, they stay put. Bacteria swim or they come together. I will give you later an example of something that moves all the time in order to survive. Well, everything is moving all the time in order to survive. That's the way we dissipate energy, collect information, and reduce continuously our entropy. When we start reducing when we stop moving, our entropy starts to grow. So that means we're dead. So in the last 10 years, I've been trying to develop techniques that allow me to measure the energy dissipated in biological systems at the nanoscale. Proteins, DNA, or molecules happen at the nanoscale. I will go back later to that on why. The reason is the previous slide that when I was showing you DNA, at the nanoscale, you're able to couple external signals, electrical, light, whatever, to a movement. A chemical reaction can couple into a movement, can couple into a shape. It turns into a computation, it turns into a shape that is able to understand the environment and potentially can produce computation. So I've been interested in measuring the thermodynamics, not yet the information. This is something I'm thinking now, how I start measuring information. I'm just trying to measure the energy dissipated in this system. So I'm interested in how we are built up from the nanoscale up. In the case of animals like us, I told you about Pierre de Jean, we are built up of cells, but a lot of polymers, again, building up this, bridging up the scales from the nanoscale to our whole bodies. So we are jelly, we're made of collagen. I'm very interested of measuring how nanoscale structures such as collagen, which makes these meshes all over our body, all our tissue, are actually there to transmit information from the nanoscale to the microscope by using polymer physics. The idea that if you have a polymeric material, you can transmit information of what is happening at the nanoscale to the macroscale just because of your shape, your polymeric material. Plants are very different to us in the sense that they don't create tissues in which you have a mesh of collagen and the cells are embedded in this mesh, but rather the mesh is outside. This is what is called the cell wall, which is equivalent to our collagen and our extracellular matrix. And it's also a polymeric material. It's another composite that makes the walls of cells, and the cells are very geometrical and they grow by turgor pressure. So you put pressure in the plant and it just grows according to the mechanical properties at the nanoscale. Of course, it has a lot of feedbacks with the biology, but at the microsecond scale, I'll come back later to that, it does actually react to polymer physics. So I'm interested in measuring this. And a few years ago, I'm going to tell you a bit to the story of this, how we measure it, a little bit of the physics about how a plant creates its shape. Plants are interesting because they stay put. They do a computation very different to an animal because they can move around to look for food. So in their case, their computation involves growing roots and growing roots able to find nutrients and producing leaves that are able to collect light. So we're interested in plant growth, the basics of plant growth. So that's a seed. I just take you to how a plant grows. We always use this plant called Arabidopsis thaliana. So it grows a root and then grows a hypocotyl and the leaves. So I use a technique called atomic force microscopy. I go a little bit through it now. So it's a nano needle that I can come here and sit it on as the plant is growing. I scan it over the sample and I oscillate it at the nanoscale. And by the movement of the oscillation of the cantilever, I can understand how it stores and dissipates mechanical energy when I form it. So it's like mechanical engineering of plants at the nanoscale. So that's what I told you. I use this technique, the atomic force microscope. So it's usually a lever at the macro scale, micron scale. And then it has a tip. And the tip has a nano-sized ending. So I can collect information about the bending of the lever with a photodiode, and I can use a laser to know what it is. I usually also use a laser to oscillate it. So these cantilevers are coated with gold, and we use a blue laser to produce oscillations of the cantilever, so we can tap on things with sinusoidal waves. And for the way the sinusoidal wave is dissipated into the material, we can infer how this material behaves, what is its time response, which is something I'm interested in. Because if you realize, to create a shape, think of the plant you just see before. You need that every component as the plant grows because there's turgor pressure. So basically pressure comes in from within and inflates like a balloon and has that shape. Now everything at every scale must respond at the right time. If everything doesn't respond at the right time, it will not be straight like beautiful plant you saw growing before. So to grow at the same time, it has to dissipate energy in the right way. So that is the non-equilibrium thermodynamics business, is dissipating energy creates your time. You dissipate more, you dissipate more into the environment, you're slower. Is what we call soft dissipative viscous. And if you are instantaneous like a crystal, you don't dissipate energy, you're instantaneous. There's no time for you. For us, time moves forward because we dissipate. We are structures that reduce entropy. Therefore, for us, time moves forward. If you're a crystal, you're eternal. There's no time for you. So it's a bit the physics. We do very simple physics. Basically, the physics that mechanical engineers do is physics of vibration, of levers, of strings. You can, solve these differential equations, and what you usually do is to use the usual tricks of physicists, Fourier transforms and Taylor series in order to simplify the problem. So we basically drive the physics of a harmonic oscillator with a sinusoidal wave, with energy dissipation in the system, which comes out as the Q factor of the cantilever. I will come back a bit more later into the details with that. So we demonstrated that using this trick and measuring these equations, so basically measuring the amplitude of the oscillation far away, the amplitude of the oscillation when we are near the sample, and the phase of the oscillation far and near, and combining it with the quality factor of the cantilever, how much energy is dissipated into the environment, we can extract the the elasticity and the viscosity in 3 dimensions of the cell. So we define the E storage and E loss. E storage is similar to the Hooke's law, the K of the spring constant of the Hooke's law, but in 3 dimensions. And this is the energy dissipated into the system. To do these experiments, in this case, what we do is to come with a lever, indent the sample, and at the same time we indenting it, we oscillate the cantilever. And by doing that, we can extract the stiffness and the viscosity. Now, in using this technique, we put it on the plant and we started measuring plant cell walls. As I said before, it's a composite, and the cellulose are amazing crystals. Actually, cellulose is the most abundant polymer on Earth, and it's a crystal, funnily enough, and it's piezoelectric. That means mechanics and electricity are coupled in it. It's much more mysterious than we want to think. And I think a lot of technology in the 21st century is starting to be a lot of research on using cellulose for all sorts of things, including transparent displays and all sorts of things. Anyway, so the cellulose grows perpendicular to the direction of growth of the plant, and is in between the cellulose in the plant cell wall. You have pectin, which is gel, is the stuff that is in your jam. So we think that the cellulose gives the stiffness, what I told you before, the elastic part, the instantaneous response, and the pectin and the other soft polymers around it gives that time response that we need to keep that shape. From a long time ago, there was an equation, phenomenological, this is not derived from first principles, that described the growth of the plant, and it has to do with a yield threshold, basically the mechanical properties in the yield and the torque or pressure. And that's the Locker equation. Anyway, so we come with our AFM and we indent and oscillate, and then we get an elastic and a viscous response as we oscillate. So this is a bit the math we did. We have to solve this equation, and for a material that will respond at many different frequencies. So depending on how fast you push into this material, you will have a different response, because the polymers at the nanoscale will respond at the microsecond range, but as you go as big as range, you will have responses at the second scale, at whatever scale. So we were interested in the microsecond scale because it's what polymer physics predicts as the mechanical response of polymers at the nanoscale. And basically we just solved that question for a system, we model the plants as linear viscoelastic materials in which you have springs and dashpots. The dashpots is the ones that respond with time. So this is where the energy is dissipated, is related to the viscosity, and these two springs, this is equivalent to an electrical circuit, but just using Kx. So these are a Hooker spring, and this is a dashpot. Anyway, so doing maths, we arrived to these equations where we can extract this elasticity and viscosity from the AFM, and doing a bit more maths. Oh, a phone went on. I'm not going to get that, but it's going to be noisy. So we can, using different linear viscoelastic models, we can extract or things like the time response, or the time response of the material, which is what we are after. Anyway, so one of the things we did, if the plant behaves like the Locker equation, it should have the shape of a standard linear solid. So this is the experiment I just described, and these are the results we were looking for. And these are maps that we work with the AFM using this technique. So we are able, using the maps of the liver and how the liver moves, to reconstruct the maps of the elasticity. So that is coming with the AFM in a living plant like that one. I'm putting the AFM there in the junction between these cells and trying to measure how the cell walls with different orientation store energy and dissipate energy like that. And then with the statistics, which is nice. This is something for biologists. Another thing we were trying to know is, from this data, can we actually reconstruct what a mechanical circuit better represents the plant? So what we found is that, doing some mathematics, that we could predict the shape of the curves of E prime versus double prime if we represent them against these. They are dispersion graphs. And plotting the data against them, we could see that, interestingly, our plant behaves like the standard linear solid, which is here, what I was showing you at the beginning. So you have two springs and a dashboard represents very well the mechanical behavior of our plant. This is interesting. The fact that the mechanical properties of our plant as it's growing follows a linear behavior is telling you in terms of the mechanics and the energy dissipating, is telling you something already about how the plant is using energy in order to create a beautiful shape that doesn't go off to any other shape. I will come back later to that with some thermodynamics calculations, but it's also interesting that recent work has shown that the metabolic energy dissipated by a yeast cell, which is very similar to a plant cell, as it grows, also is linear. The way it dissipates is with time is linear. So there seems to be a correlation of the way energy is used on the plant with the mechanical design of the plant in order to create their shape. I need to think more about the physics of that. But from there, we can extract already interesting quantities. And what we were interested is growth. So now we measure the mechanical properties of plants, energy stored, energy dissipated. Does this correlate with the speed of growth? Because that would start to give us really the link between mechanical properties of polymers and the shape of the plant, and indeed it correlated. When we submitted these papers, I don't know why the biologists were very skeptical about it. We have to do a lot of controls, and we use mutants. So we use plants where we remove some genes so they don't grow so happy. And we start to see that really the way the plant grows is very much related to the mechanical properties at the nanoscale, as you would have predicted. But anyway, this is part of the work. So this is a slide I put for biologists, because biologists would not understand why, if the plant grows in the seconds and the minutes scale, why the mechanical property that you measure at the nanoscale in the microsecond range can be correlated to growth. Why should they be correlated to growth? Why they're interesting? For us, it's pretty straightforward, because polymer physics theory tells you that the times that are relevant for a polymeric material are related to the radius of duration, basically related to the size. And indeed, for something like the polymers inside a plant cell, we would expect that the microsecond time scale is irrelevant. Again, this goes back to the idea that when you have systems that are hierarchically constructed from the nanometer scale with soft matter, the time scales at different scales are different. I think this is quite intuitive for physicists, but biologists like to think of as slower processes. In fact, there are slower processes in plants. So there's some enzymes that are released in the second scale to produce shape as you go along. But I think those are probably there to diffuse, to release tension as you grow. At higher time scales. Again, my phones are just going all over. And I can't tell, I don't know, someone is really trying to reach me. Okay, not the right time. So anyway, what we wanted to create was maps of the mechanical, of the time response of those cells. Again, we realized just doing algebra that we could relate the time to the derivative basically of E prime over E double prime. That means the gradients, which is very interesting, of the energy stored and the versus the energy dissipated. So we look for those gradients, and then from those gradients, and doing a bit of math, we were able to create, and we are very proud of this, the first maps of time. Here's the time. These are 1, 2, 3, 4 cells growing together in a plant. And these maps are actually giving you the time response of the different components. So that's, you may think it's boring, but it's quite exciting for us. And just using this model. I show you some pretty pictures we took of leaves. Leaves have this beautiful structure. So they have these pavement cells. So depending on where you are, these ones were very cylindrical because they need to get that shape. in order to get the leaf shape, plants create these pavement cells. And in between the pavement cells, there are stoma, which are these pores that open and close when the plant wants to get more oxygen or remove some water. So these are sensors of what is going on around them. And together they create this mechanical structure that creates the leaf shape and is mechanically active. It doesn't look like it, but if you look at the micro scale, this stuff is moving all the time. It's responding. It's a very clever computer. Again, it's responding to the environment to open and close. And with our technique, basically, we are trying to understand the time responses of these structures. And in the future, I really want to look into the computer. I want to try to understand how the opening and closing of the stoma actually relate mechanically in order to create shape. Remember, the leaves move according to the sun. There's feedback controls there that are orienting the leaves to energy. My dream is to basically try to understand that computation and integrate this understanding of the mechanics at the nanoscale, but at the moment we don't know it. And my job as an experimentalist is to create techniques that links with theory so we can know it in the future. These are roots. Roots are different. They look for nutrients. They have different mechanics. They have different shapes. And this is also something I'm interested in. Let me check the time. Because I've been talking a lot already. Let me go to my living thing. Quickly, I take you to a little bit of the thermodynamics we are doing. So we're not happy with the phenomenological equation of growth that they had given to us. We wanted to derive our equation of growth of plants from first principles and for non-equilibrium thermodynamics. So we use something called the reciprocal relationships of Onsager. Remember that was in my slide about the history of physics and complexity, which tells you that even there is a slow variable in the system, something that moves fast and the other things move very slow. The fast variables are near equilibrium and one can use non-equilibrium, just a perturbation on the energy to calculate what is going to happen to the fast variable. So using the Onsager principle, actually, we could derive the equation of growth of a plant. And one of the interesting predictions that we got from using thermodynamics to derive the equation of the plant was to relate, that it would tell us that the viscosity that we were measuring in our experiments could be related to the velocity of growth. And indeed, our experiments correlate, fit very well with the predictions we made with non-equilibrium thermodynamics. And it tells us that environmental conditions must be stable over time scale of growth in order for the plant to have its shape. So I'm going to skip this because I want to take you to another example. Sorry. And I don't have water either. So I've been telling you about how plants create their shapes, and that my dream is to understand the computational power that is embedded in those mechanical structures, for example, in the leaf when it's looking for the sun. So what is going on in the leaf to be able to translate the information that is getting in the sun, from the sun, basically energy, that in orientation, because of the shape of the leaf, it will be able to get the light in different orientations in different ways. So there's gradients on the leaf that will produce a mechanical rearrangement. The thing is, with plants, they cannot move, but organisms that can move make the computations in different ways. And I want you to present you with this example, because a big part of the biological physics community, bioinspired computer community, thermodynamics community is starting to study this mold. Maybe you already have heard of this. It's called the slime mold. So it's a mold that rots wood and it's pretty amazing. If you work with it, it just walks around and finds food. And in order to find food, creates networks of tubes. So there's a lot of tubes within it, and like little tubes of a structure that has fluid inside. And the tubes find the food and create networks. And so it's an absolutely amazing computer that is able to optimize. So all these optimization algorithms that we have now for machine learning, this thing just does it using gradient and structure. So I'm going to play you this video. These guys in Japan show just an example because it's pretty amazing. They put food on the train stations of Tokyo. So basically they made an imaginary map of Tokyo underground, and they put a little bit of food for this mold on each of the stations, and then they let the mold find the route between them because of this capacity the mold have for optimizing the best route. I guess they also put some geography in it. So basically they put the mold on some geography that looked like Tokyo. And shockingly enough, there's the mold growing and finding food and starts creating these tubes, and these tubes optimize the path to create, to extract the nutrients from that food. And then they let they let it go, and they end up with this structure that basically looks like the Tokyo Underground subway. The Tokyo Underground subway also is optimized by the geography of Tokyo. And these guys managed, the mold managed to optimize that by itself. So yesterday came out in PNAS a beautiful paper that I wanted to show you. I haven't had too much, I don't know what, this is not moving forward. Okay, here's the paper. Oh no, I managed to move forward. So it just came out yesterday. This trying to understand what is the computation that this mold is doing in order to optimize the intake of food. Again, think about a biological shape created by evolution, being able to create, store information, and create a computing system that allows you to be alive for a long time. This is what I told you at the beginning. How are you able to be alive if you are very soft in this universe for long enough? By making a computation. So this is how this guy does it. So this is the title of the paper already tells what they found out. Memory of a nutrient stimulus position is encoded in the network hierarchy. So this is a very computer science type of doing this. So these guys are Mirna Kamar and Karen Alem from UPenn. So they got this Physarum polycephalum, which is this slime mold. And basically what they say is that the network-like body plan of the organism is what the tubes of varying diameters is what makes the network. And the organism responds to where the food is. So this is the mold, and they put food here, and they over time the mold finds it and moves it and is able to move there in order to eat it and extract energy from it. So these guys, they look at the network and they look at the diameter of the tubes, which is very clever, near the food and far away from the food. And they try to make a model of why and how this thing is able to change the diameter of the tubes. And that is the way it creates a memory and the computation itself. So, so basically memory encoding, it is encoding memory in the structure and the size and the diameter of the tubes, which is pretty amazing. So this is what I just told you before. They put some food and then what they find to start with is that the tubes and near the food are thicker. They have a bigger diameter than the ones far away. How does it do it? So it looks like the tubes are receiving a lot of a softening agent to grow in diameter at the expense of other tubes shrinking. And so basically the tube's capacity for flow-based transfer get upgraded towards the nutrient location, redirecting future decisions and migration. Again, here we go with this idea of time, size, and structure, always linked in order to find. So the idea that you go there and you make your tubes bigger is also giving you the time response of the system and how that propagates. So these guys, basically, they cut it to a shape, because this thing you can cut it to a shape, and they put some food, and then they measure the diameter of the tubes near the food, what I just told you before, and you can see this a thickening here of the network. And then they measure the diameter of the tube as you go away from the food, and basically the diameter of the tube increases near the food. And then here's the theoretical model, which is very similar to the theoretical model I use for my AFM experiments. So because they have the Stokes equation, because there's some fluid going inside the tube. I haven't had time to study very well this paper because it just came out yesterday. So, and they're related to the contractile stress and the elastic stress. So basically they are linking mechanical properties, like I was telling you before, energy stored, energy dissipated, elasticity, viscosity, in this case, stress, and cortex with the flow within, and then solving, and then what they put is that they think there is a gradient. Some gradient is driving the agent that loses the tube, changes the tube mechanical properties and let it grow into one direction to make it thicker, is a gradient. There's some agent that is doing that away from the food, and they model it. And from this model, in which you have a distribution of that agent over time, and then they predict that the tubes nearby, which will be here, will have this behavior in terms of diameter, and is totally driven by the distribution here going here. So that's why you have a Stokes equation. You just have fluid dynamics going there. And this is experimental results, which is very clear. An interesting finding of this paper I was looking for all the time was what is the losing agent? They don't know. They think it might be ATP. They don't know really what is, something is released near the food that is changing the diameter of the tubes, but they don't know what it is. So they have a good mathematical model, but they don't really know what is causing it. I'm just going to finish with what next. This is something I gave the other day in the talk of the physics department about. So I just show you how, as biophysicists, we're trying to understand the link between information coming from the environment, mechanics, a structure. In the case of a plant, the structure is this multicellular thing with a lot of cells that responds to the environment and interlock with each other through mechanical interactions as well as other interactions. In the case of these organisms, I just showed you before, very simple organism is just able to create a structure that changes radically depending if it has food or not, and doing these networks is able to produce a computation that is able to optimize the path to obtain food, which is pretty amazing, and I think it will be used in the future for a lot of things. Another area that is becoming interesting, I'll go very quickly. You heard about quantum biology, the idea that quantum is somehow, quantum effects are important in biology. I may just tell you a little bit what Roger Penrose thinks, and what some of us are thinking of experiments we can do at the nanoscale with biological systems to understand a fundamental problem of physics, which is what is the step between when quantum becomes relevant in the classical world. So as you know, quantum computers these days, some people working on quantum devices are trying to understand things like how does quantum fluctuation, a colleague of mine is working on the materials, how does quantum fluctuation translate into a mechanical movement? Roger Penrose thinks inspired by Goodell. Remember, I started my talk talking about Goodell on things that cannot be computed classically. So Goodell tells you that there's some reality that cannot be found with an algorithm in the classical sense of Turing. And Roger Penrose thinks that our brains are not a classical Turing machine, and that artificial intelligence of the kind we have, like human-like artificial intelligence, is not a matter of putting complexity into the system, into the machine learning. There's something else. I put here this picture. This is from a sculpture friend of mine who makes these sculptures. And these ones are from a park that he put these sculptures in Tokyo. And immediately when I saw this sculpture, I realized that it has the same dimensions of my plant. So he had the intuition of the shape of a plant. Actually, when you see it, you can see he calculated a kind of shape. That's why it's interesting to see it in the park. So there's this human intuition coming up in art and also in physics about why these things have the shapes they have. I incidentally also show you that my plant also has intuition of mathematics. I mean, I told you that the leaves are able to find the light, and very interestingly, and we know this from the Greek times and before, you do this by creating magic angles. So my plant also has a magic angle here. It's also intuiting mathematics. So Roger Penrose thinks that the reason why we have things like intuition, or we can understand infinite, like Cantor. Imagine Cantor when he goes and says some infinites are bigger than others. The idea that we can, and we cannot compute infinity. This is one of the ideas of Penrose, who himself had worked a lot in infinity and trying to understand why we can understand infinity with mathematics. He actually does it with drawings, which I think is part of the way he's so good at realizing this link between intuition and computation, because of his Penrose styles, for example, right? So he thinks that is some quantum computation involved, and that the parallel power of quantum computation would allow us to have things like intuition. So I'm very interested in this interface between quantum devices and biological computations at the nanoscale. The idea that at some point the quantum collapses into classical, and that is important. I don't know, we don't know why, how. So my friend in materials, Natalia Aras, for example, is working on how quantum fluctuations affect the bending of the carbon nanotubes. And we're now thinking of understanding, so creating similar thermodynamic systems in the quantum realm, and in the just basically copying biological systems. So with my technique, I can measure energy dissipation, and they can measure, and we want to see how the energy dissipation in a quantum system and the energy dissipation in a classical system, that is a protein, are related. So for example, by creating Maxwell demons in the classical and in the quantum, we're exploring this. And eventually we want to do experiments that link biological structures, potentially quantum effects with mechanical changes at the nanoscale. So basically, the biological shapes might be also interesting to understand how a quantum computation can stay at high temperatures. One of the main problems of quantum computers is that they have to go at very low temperatures, because in vacuum, like this nanotube, the nanotube dissipates energy into the environment, the quantum information is lost. But you can imagine a biological system, and there is some evidence that this might be happening, that evolves over time so that it controls the energy dissipated into the environment to allow the quantum computation. So this is something I'm interested in exploring myself. I quit here the presentation, and we can have a bit of a discussion if you don't want to run away too quickly. Okay.
Thank you so much. I'm so happy that we got you for our 6-week talk, because this is yet another thing that you know, it's so easy as an undergrad to go through your whole degree and never get exposed to this sort of thing, and not realizing that that's something to go into when it would be a great fit for so many people. So thank you so much.
I think that's what I want to do, and also bring a bit of history of physics in the whole story, because it's also easy as an undergrad to lose track of why we arrived to the point we are now.
Yeah, absolutely.
So you can unmute yourself. Okay, let me see some of the things on the chat. Definitely you unmute yourself if you want to make a question. Word measuring biological time used in the title of what does biological time? I think I explained it. Relativistic effort. Okay, so what I meant about biological time is a mechanical time, the time response of my structural material when I deform it. It is important, this mechanical time, if you think about it from the computational point of view. I think it came clear in maybe more clear in the moving mold. So the capacity to understand the environment, like the leaves of the plant. So there's information coming to you, and then you need to restructure yourself in order to get light, are determined by the mechanics. And this is the time I want to measure. It's just a mechanical time, but it's deeper if you want, because that mechanical time has to do with the energy dissipated into the environment. And that energy dissipated into the environment is already a deep physical quantity. It's the non-equilibrium thermodynamics that is driving the very construction of the biological system. So it's not relativity, time. These are not relativistic scales. They are thermodynamic time, if you want, in these scales. No, that's the time of life. In a way, the time of life is determined by the fact that we emerge from polymers. That is the time of life. The soft matter time, that radius of duration of polymers, is important, because the reason why biology emerges at the nanoscale is because the scale where you can couple information at that time, the microscale. And that's why it's interesting to go to quantum, because those would be shorter times. So it has to do with the way we process information, with the computing power of biological systems. All computers are clocks. You need to have all these times in order to make the computations. Biology evolves by creating the clocks. That's the time, not relativistic clocks, thermodynamic clocks. But maybe you know more physics than me, probably. Maybe you can come up with some other ways, and you can see this time.
No, as I've wrote in the chat, I think that's we're on a completely different scale. Relativistic effects are completely negligible.
I know. This is a different thing. But yet, if you see, there's another convergence I didn't talk about. So we have this idea of general relativity doesn't match with quantum mechanics, was still there. And again, this collapse of the quantum world into the classical world, the beginning of gravity, when something starts to have gravity, when you start with a quantum system, it looks very far away from life, but it might be the same physics that makes life to start. If we need a quantum computation in order to create a biological molecule, then there's another beautiful convergence there between different areas of physics.
Yeah, it's an exciting time. We had a talk on quantum computers at some point. If people want to go back, I believe that one's also on the YouTube channel. So go back.
Actually, I did invite my colleague in materials if she had time to give this seminar with me, because she does quantum devices, and it would have been fun to prepare it to both of us converging. Maybe we can do that another time. So quantum biology meets quantum devices, because I think that's next convergence.
Yeah, something I always like to plug whenever quantum computers come up is that IBM not just does a lot of quantum computing on their side, and they have quantum computers themselves, but they have a really great outreach program called Qiskit, where if you're into it, there's so much material there about the fundamentals of it, the physics of it, but also biological, like chemistry computations, the computation side of that. They have a great online community. Do check them out, and you can run your own code on their quantum computers for free.
Yeah, I think this thing, as I show you now, of this little mold able to produce computations just by shape, this type of biomorphic computation, natural computation, is going to be really influential. The idea that you can evolve your algorithm depending on what you're feeding your algorithms, I think there's going to be so much cross fertilization between biological computing, biological physics, quantum computers. Is the time of, we're closing to this dream of understanding why intelligence emerges in the universe, and that's why biology is a physics question. Is the main physics question actually. If you realize, what Einstein, I was reading this wonderful book I recommended. It's called Einstein's War, about how the social environment also affects the theory of general relativity. So when Einstein is looking at the data of time, etc., that he came up with, things that how we think we are, basically it was a how do we understand question. What is the relation between our intuition of time, the experiments we make, and the logic and the reality? This is at the heart of why he started thinking that there was something wrong with the way we were interpreting the universe. I think intelligence has always been central to physics, because physics relies on intuition, and not only logic, intuition. I mean, this is Richard Feynman's famous intuition, how you create a physicist intuition. This has always been part of our identity. The thing is, now intuition itself is becoming part of physics, which is great.
Yeah, I always recommend, if you have the extra time on the weekends, go back, read about the physicists that did the stuff that you're interested in, and you'll be surprised, like the things that they were thinking about 100 years ago as well, and knowing about the circumstances under which they were working. It's such a fascinating story.
Okay, people are already leaving. I have to leave too, because as you saw, I had a lot of phone calls that I couldn't attend.
Right. Thank you
Okay.
so much.
Yeah.
I don't think there was anything super important in the chat. If you always reach out to the speakers if you have.
Yeah, you can send me emails if you want as well. I'm sure you won't, because you're very busy with your term, so am I. But anyway, hopefully we can meet maybe next year.
Yeah, absolutely.
We might, reach out if you see me, and when you come back to the apartment, or send me emails if you want to discuss more about this. Maybe after, when we finish the term and you go home. I'm happy to discuss.
That'd be amazing. Thank you so much.
Thank you
do check if there's any fires and your phone calls.
Yes, I know.
Thank you so much.
Bye. Lovely to meet you both. Thanks.
Same. Thank you. Bye. Brand. I'm sure people are leaving. Thank you so much for coming. If you're still here, I put a link in the chat for people who found this interesting. If you're interested in how shape emerges and stuff, it reminded me of an episode from the Sean Carroll podcast. That was one of the more recent ones. I put it in the chat if you want to go check that out. If you don't know his podcast, it's generally a very interesting one. If you like these talks, you'll probably like his podcasts. And check that episode out if you find this particularly interesting. I thought it was a really interesting one. So thank you to everyone who stayed until the very end. And hope to see you next time. Bye, everyone.